Linkage? Linkage is the easiest genetics topic I'm going to teach you yet. So you have this page out. Linkage essentially refers to when you look at a chromosome, there are many different genes on a chromosome, and we've talked about that before, correct? So if we were to draw out a chromosome, for example, all along here would be different genes. And remember, chromosomes are made up of DNA, right? So this is a long strand of DNA that essentially makes up the chromosome. This part of the DNA would be referred to as one gene. This part of the DNA would be the next gene. So essentially one gene codes into the next one. Now, where genes are located on the chromosome affects how they move during the process of mitosis, during the process of meiosis. So when crossover occurs and they're exchanging arms, where these genes are arranged depends how frequently they will cross over. And what linkage is about, and if you want to look at more detail, I have a PowerPoint you could take a look at that talks about the history of Thomas Morgan, who was the first person that he was dealing with fruit flies. And instead of getting normal ratios like the 3:1 or the 9:3 to 3:1 in a dihybrid ratio, all of Mendel's traditional things, when he took the different traits, he would get extremely lopsided ratios. So instead of a 1:1 to 1:1 ratio, he was getting 2:4 to 55 to 42. And he said that doesn't actually match what Mendel is saying, which is what everybody was believing in at the time. So when he did more research, he found out that those genes that provided those really lopsided ratios were really on the same chromosome. And how often they crossed over or exchanged information depended how far apart they were. So the whole idea of linkage means that genes are linked on the chromosome. So any of the genes here would be linked because they're on the same chromosome. As opposed to if I drew another chromosome over here, genes over here would not be linked to this chromosome because the chromosomes can move independently of each other. Does that make sense? So if you want to jot anything down at this point, linkage refers to genes on the same chromosome. So the key is linkage refers to genes on the same chromosome. So essentially what Thomas Morgan found is that the further apart the genes are on the chromosome, the more often crossover is going to happen and those genes will be switched with the other chromosome. So it's going to create more variation. So essentially the further apart a gene is, the greater the chance that crossover is going to happen, which means more genetic variation. The closer two genes are together, the chance that a chromosome is going to break and cross over between those two genes is pretty low. Whereas if you look at what's the chance of a breakage occurring between these 2 points, there's a greater chance because it's further apart. Okay? So that's all that linkage means. Now, the whole reason you might want to find out if genes are linked is so that if I gave you 5 different genes and I gave you some information, you'd actually be able to draw a chromosome map and say, gene A goes here, B goes here, C is somewhere in the middle. You can actually kind of visualize the order of those genes based on the rates at which they cross over. So making a gene map, so if this was a chromosome and saying, here's the information and plotting them out, is what Thomas Morgan was trying to do. He was trying to figure out where on the chromosomes are these genes located and how often do they cross over and what does that mean for the offspring that are produced from that. Now, here's why linkage problems are the easiest type of genetic problem to do. There are only 3 options. So the questions will always say we're going to take these parents, we're going to cross them, we're going to put that in a test cross, we're going to cross it with something that's homozygous recessive, and then it's going to give you a set of information. And from that information or those offspring, those ratios, you have 3 options to choose from. So if you look at the offspring and there are only 2 types, it's referred to as complete linkage. So if it said there's a black tall and a short, so I said black tall and a brown short, there's only 2 types that are given, you would say that that's complete linkage. That means that they're on the same chromosome, there is no variation, they are not crossing over. That's what you will get time after time. Essentially, they're so close together, nothing will ever cross over in between them. So they will always travel as a pair. The other way to double check this one is when you look at the offspring and you read the question, those offspring will be exactly what it tells you in the question. They're exactly like the parents. So that's the 5-second question solve. Then we get into, if there are 4 types of offspring, there's 2 options. The first one is if you have a 1-to-1-to-1-to-1 ratio or a percentage of recombination that is 50% or higher. That tells us that it's independent assortment, which is a fancy way of saying that they're on separate chromosomes. Every one of the genes that Gregor Mendel picked was on a separate chromosome in that pea plant. So he was fortunate that flower color was separate from, or was on a different chromosome from the height of the plant. So he didn't have any strange ratios. Everything always worked. Now, if you have a lopsided ratio, so there's 4 options, but they're not that 1-to-1-to-1, and it's lopsided, or you get a percentage of recombination, which is 49% or less, that's referred to as incomplete linkage. Now, that means they are on the same chromosome, but sometimes they cross over. So sometimes you get some variation, sometimes not so much. So essentially, you read the question, you look at the parents, you check the offspring, and the first question you ask is it 2 offspring or 4 types? If it's 2, you know it's complete linkage, you're done, you move on to the next question. If it's 4, you check the ratios and the percentage, which is what I'm going to show you how to do next. So, the key here, you need to underline the parents as you go, so you know what they look like, so you can pick them out when you get to the offspring. So step 1 says, underline the parents in the question. A certain plant is tall and has red flowers. So there's parent number 1. Another plant of the same species is short and has white flowers. So there's the second parent. There's the first. 2 pure breeding lines were crossed and all produced tall plants with red flowers. A test cross, so they're crossing it with something that's homozygous recessive, yielded the following results. So, first of all, are there 2 types of offspring or 4? Okay, so there are 4 types. So now I'm dealing with, am I getting an even ratio or a lopsided ratio? Can we tell for sure here? Okay, so when you get a series of numbers like 78 to 80 to 65 to 76, they're maybe pretty close to 1-to-1, but you need another way to confirm it. So what we're going to calculate is called percentage of recombination. In order to calculate that, we have to figure out which ones are the recombinants, so which have a trait from each parent. We're going to add them up, divide by the total number. We'll get a percent, and that will tell us 50 or higher or 49% or lower. Now, here's how you figure out the recombinants. We highlighted the parents. So first thing I do is I go to here, and tall red, does that match one of the parents? Okay, so I would label it with a P for a parental type. So it looks like it's parents. Then short white. So there's my other parental. Therefore, the recombinants would have to be what? The other 2. So these ones are the recombinants. They are a recombination of both parents. Now, often they are the lowest numbers, but not always. So always identify it from the question. The other way to check to make sure is that each of these is kind of like a reciprocal pair. If this is tall red, then this has to be short white. You see how that kind of complements each other? And if you have tall white, the matching has to be short red. So your second double check is make sure if you have a tall white, that you have a short red. So I guess our first checkpoint is, are you okay to identify the recombinants? Okay. So we've identified the recombinants out of the 4 choices. Step 3, we're going to take these two and add them together. So 65 plus 76. So we add the recombinants together and divide by the total number of offspring. So you need your calculators. Or if you're really good at mental math, that's perfect. And then I need the total number. And I just need some people to double check with me to make sure I've entered everything in right. Okay, is the total 299? Did I add that correctly? So we have 141 over 299, is that correct? Sounds right, or we know for sure it's right? So to recap, I added the two recombinants together, so I get a total. The number I divide for is all of these added up. Now what I'm going to do is take a percent, so 141 divided by 299, that equals 0.47. We can go to 3 decimal places here, 472. And if I multiply that by 100, what's my percent? So 47%. You don't need to show me that you can write it as a decimal and then convert it to a percent. You can simply go straight to percent. So if we are at 47%, we go back to here and we say we couldn't decide if it was the 1-to-1 to 1-to-1 ratio or the lopsided ratio. Now, if it's that independent assortment and these are truly on different chromosomes, my percent of recombination will be 50% or higher. Is it higher than 50? So that means that it's the lopsided ratio that's 49% or lower. Therefore, these two genes are incompletely linked. So you would say the percent of recombination is 47, and that means incomplete linkage. Question's done. Your evidence is the 47%.
That's it?
That's it.
That's all?
And there's only 3 types of options. Now, what this essentially means, that if we were to put the gene for, let's see, height, because that's tall or short, and the gene for color, these two genes would be 47 map units apart. Or if you're a famous scientist, you can name things after yourself. So what do you think Thomas Morgan called them? Close.
Thomas Morgan.
Centimorgans.
Oh.
So if you see anything that CM centimorgans refers to, right, that distance apart. So on a chromosome, now centimorgans, that's not direct translation into centimeters apart. It's that's like a map unit is a way to provide a perspective proportion for that. So they are 47 map units apart. Crossover is going to happen quite often, but not all the time. So sometimes you'll get a greater variety of offspring produced. Sometimes you won't.